Hey, this is Joel Duff. Welcome back to the channel. Been a while since we've had an episode of the Hemoglobin Challenge. And so, episode number 9. And I've got two items for this episode. One is I'm going to give credit to a challenger, somebody who emailed me and sent me a paper as a response to the Hemoglobin Challenge. And I'm going to provide my response to that here. And we also have Brian Thomas, who's from the Institute of Creation and Research. He comes close. He comes close to passing the hemoglobin challenge, but he has to toss in a couple extra words, which gives him a failing grade. But let's give him a little bit of credit for being slightly more careful with his terminology this time, and maybe next time he'll pass the hemoglobin challenge. So we've got those two items coming up next. Before we get to Brian Thomas, let's first review what the hemoglobin challenge is, since it's been a while. It's my challenge to anybody who has inappropriately said or implied that dinosaur bones have unfossilized red blood cells or undecayed hemoglobin in them. And if they recognize that error and describe this soft tissue more accurately in the future using terms such as hemoglobin-derived porphyrins or chemical breakdown products of hemoglobin or evidence of former presence of hemoglobin, those are suggested terms you might use rather than talking about fresh blood or blood cells being found intact. Some similar phrase or sentiment will give you a lot of credit from me for passing the hemoglobin challenge. So let's see how today's contestants have fared. We're going to start with Brian Thomas at the Institute for Creation Research with his article, Dinosaur Blood Vessels. Actually, it's not just Brian Thomas, it's also another co-author at Institute for Creation Research, Frank Sherwin, but we're mostly focused on Brian Thomas since that's his soft tissue preservation is his thing. All right, we don't need to spend a lot of time on his very short article, which is just a description of a new paper that has come out by Mary Schweitzer or Dr. Mary Schweitzer and some of her colleagues and students, looking at her latest results, looking at soft tissue preservation in dinosaurs, for which she's famous. And in that, they're reporting about using a new technology to look for remnants of protein residues and materials. And so here we have this quote, their results support the presence of epitopes of a heme-containing compound consistent with hemoglobin. So again, so here Brian Thomas is reporting what was found by Mary Schweitzer, and he's actually reporting the words right from the article. So I have no complaints about his reference to hemoglobin here. And then down here, a little while later, he says, since the hematite in these dinosaurs was bonded to blood proteins, they confirm that it came not from the surrounding rock, but from dinosaur bodies. In fact, a 2025 study confirms partial hemoglobin preservation in dinosaur remains. Again, this is an appropriate way to reference what was discovered. All right, and this is his words, and as he's describing a 2025 study, and he is properly citing the fact that they found partial hemoglobin preservation, not hemoglobin as this really large macromolecule that contains thousands of amino acids, but remnants of the heme-containing compound, which is just a little sub-portion of the hemoglobin molecule. And specifically, he's not saying blood, right? They wouldn't find blood cells in this dinosaur tissue. Oh, wait a second. He then has to take this a little bit further. He couldn't just leave it there. He has to make commentary on this. Quality Science has again confirmed original biomaterial in these fossils. Yes, I agree. There are original biomolecules found in dinosaur tissue. Original biomolecules means that the actual molecules that were present in the living tissue, some of those molecules are still present, often in a highly modified form, but nonetheless, they haven't been completely replaced, right, by other material. But notice the next line, right? But prior assumptions, again, have crept in, affecting interpretations. Is this dinosaur blood? Raman results suggest yes. Has it lasted tens of millions of years? Only if one assumes before concluding it. So then he goes to hey, is there dinosaur blood there? Well, Raman results suggest there is. Now, this isn't wholly inaccurate, but here's my issue, and this is the hemoglobin challenge, is that his audience, all right, those who are young earth creationists who are seeking knowledge from supposed experts, right, expert scientists that are supporting a young earth creationist viewpoint, read this and they see this. Oh, was there dinosaur blood found? Oh, yeah, blood was found. What I'm asking is that they say what was really found, which is there's evidence of the remains of molecules that are part of the original blood cells that were present. Red blood cells are not actually found there intact. And blood actually refers to not just the actual cells itself, but blood is basically almost like a tissue, right? It's the plasma, it's the red blood cells, it's the white blood cells, that mix, right? That's what blood refers to. Did they find blood in those things? No. They found evidence that blood was there. All right, I'm being repetitious once again. Come back to this point in just a moment. Let's move on. Actually, let me show you the original paper. Resonance Raman confirms partial hemoglobin preservation in dinosaur remains. And notice that first line from their paper. I'm going to be fair, and here's what Mary Schweitzer and colleagues have said. Still soft. Yes, these tissues, all right, that are found in actually we'll talk about the blood vessels in just a moment. Hollow, flexible structures, morphologically consistent with blood vessels, right? Not that they're complete blood vessels, but they're consistent with the partial remains of blood vessels. We're going to look at another paper in just a moment that's very similar to this by the same individuals looking at blood vessels. I wanted to pull one line from this paper that Brian Thomas doesn't actually use, but I think it's an important thing to remember for our next discussion about another person who sent me an email challenging me. This line right here. All data support the hypothesis that these tissues are endogenous to the once living dinosaurs, meaning that well, I don't like to use the word tissues because it makes it sound like there's whole cells that are preserved there, but the remains of organic biomolecules present in structures that maintain some of the shapes of the original cells or tissues that were there actually do contain some of the original atoms of the dinosaur itself. And that's what they're discovering and trying to examine and understand and characterize in these papers. So then the next sentence here highlighted in blue becomes important. Here we conduct further studies on vessel-like materials. Vessel-like materials. That's actually what is being found, or it's an apt description of what they're looking at. It's vessel-like, meaning it has the shape of vessels. It is got some flexibility because of the molecules that are making up that particular shape are bendable, right? They're not crystal lattices, right? They're made of atomic bonds that are able to shift. And they are materials, right? Not cells, right? Not whole cells that make up tissues, the tissues that make up the blood vessel wall, but the material that's left over from the decay of the vessel wall that still maintains some sort of shape. Well, that is the vessel-like material, right? And then notice what they say, like hereafter vessels, right? Hereafter, we're just going to call them vessels throughout the rest of the paper. So it's totally understandable from a non-scientist point of view. When they see headlines, especially in the popular press, blood vessels found. And again, it's kind of like saying hemoglobin is found, but actually hemoglobin as a whole molecule wasn't found, but only a very small portion of it there. And that portion tells you that hemoglobin was present in the past, but most of it is decayed and left a little tiny stable element behind. This is like the vessels too. The vessels have most of the components of the vessels have decayed and disappeared, but there is a portion of the material of the vessels, specifically the collagen fibers, they become highly modified and stabilized and retain the shape of the original vessel that was present. And so we can pull these things out of dinosaur bones that are the shapes of the vessels that were there, and those shapes contain some of the original biomolecules. Okay, now with that, let's move on. One other thing I just remembered, since I've been talking about hemoglobin, I've put this picture in here. So hemoglobin is a very large macromolecule consisting of hundreds of individual amino acids. Each amino acid contains 30 to 40 atoms. And then embedded in a hemoglobin molecule are the heme units. And the heme unit is this very stable cyclical carbon compound with iron in the middle. And that particular compound will be modified somewhat as the rest of the hemoglobin, decays, right? All the other amino acids decay, and you're left with these little heme units, right? And one blood cell can contain millions, hundreds of millions of hemoglobin molecules, right? Each of which has thousands of atoms. And so we're left with just a few of these heme molecules that are still present in these dinosaur bones. And they represent the fact that there was blood there at one time. Not shocking at all, right? That there was blood in, actual blood vessels and blood cells, red blood cells with hemoglobin in them at one time in living dinosaurs, and that some of those biomolecules could have been preserved over time. So that brings me to an email way back in April 5th, 2025. I had a short email exchange with an individual and was explaining some things about things I had said about red blood cells. And so they came back to me in an email that I completely missed. I went back to it because somebody else reminded me of this recently. I searched my email and discovered that I hadn't even opened this particular email. I apologize to that particular individual. And here's what they said. They said, I do want to challenge you, though, on the no blood vessel claim. So this is after a discussion of the hemoglobin thing. So the blood vessel thing is also, kind of fits in with my hemoglobin challenge, even though it's not as specific as like the things I talk about with hemoglobin. I do think that it's incorrect, but please tell me if I'm wrong here. I submit the paper below to you, which says in the actual title that they found blood vessels from a dinosaur. Mass spectrometry and antibody characterization of blood vessels, all right, from this particular dinosaur. As you read the pair, it seems pretty clear to me they're talking about actual blood vessels. I have more to say, but I'll wait to get your feedback on this. Look forward to your reply. Here is a partial reply because I'm sending an email as well. So here's what I want to say about that. And again, I think it's completely understandable that because, like he says, the actual title says blood vessels. But when you get down right to it and you ask yourself, well, what is actually present there? Well, I've already just talked about that. I've said that there is vessel-like materials. In other words, there are components of the original vessel that are still present and still take on the shape of those vessels. But a blood vessel itself is a complex tissue that includes embedded cells and cells that then excrete material to form this matrix that the cells are inside. This really simple diagram over here shows that you have collagen fibers. In the very inner circle, you have endothelial cells. Those would be individual, like actual cells lining the inside of the vessel, cells on the outside, and then you have various fibroblasts. You have elastin. You have a number of these different long fiber chains, which are proteins, right? But they're not. But they're long proteins attached to each other. But they're not like all cells, right? Sometimes they're excreted, they're excreted substances, all right, from cells that form this matrix. That stuff is, collagen is flexible. And that's what gives you a bit of flexibility of your blood vessels to expand and contract. So now what you see in this dinosaur up here is when you eat away all the other bony material and you leave just these proteinaceous fibers, these fibers are still intact enough that they are forming the structure of the They're still forming little tubular structures of where the vessels were, and actually are part of what the vessels are. I mean, they literally are, biomolecules that were in the dinosaur that are still present and still take on that form. Now, again, what I'm going to say is that, young Earth creationists will often point to this and say, look, see, there's blood vessels. Again, it's not exact. It is blood vessels, but they are highly modified. How do I know they're highly modified? Because Mary Schweitzer and colleagues have been studying these in incredible detail in years, and they're trying to find little bits and pieces of protein to show what proteins were present in the cell. So here's that paper. This is actually from 2015. This is the one that was shared to me by this individual that has the name blood vessels in the title. And in that paper, they're talking about, so in that paper, she's reporting on the various types of protein fibers that they've been able to identify that are present in these blood vessels using mass spectrometry. And they are things like the collagen fibers and elastin fibers and a little bit of myosin, which is part of the muscle fiber of a vessel. Right? That really is quite amazing, right? And tubulin too. I forgot like that. So that's really incredible to find little pieces of those proteins. But I want to be very clear to note that what's not found there are actual cells, right? Like whole cells. Because there's no plasma membrane. That's the definition. That's what defines a cell. That's the boundary of a cell, right? The plasma, the bilipid layer, and it would have many, many different proteins embedded in it. There's hundreds of really common proteins that are found in cells. And so a blood vessel would have epithelial cells on the inside. They should have lots and lots of different components, including DNA, ribosomes, all these other structures that are found in cells. What is found, what is remaining are just a few individual proteins, of which there are large amounts of that particular protein. But these are the types of fibers that are very resistant to decay. Right? They're the kind of fibers that once they get cross-linked, and Mary Schweitzer's done all this work with iron being present, but of course there's going to be a lot of iron present because of all the blood cells themselves, right? The red blood cells that are decaying and breaking apart. And there's millions, right, of hemoglobin molecules in each one, which means there's millions of iron atoms in each of those. And those iron atoms then will bind to the actin, the myosin, and the fibers and the collagen protein, right? Producing a very stabilized yet modified versions of those proteins. And then everything else is decayed away. And so that's what's creating this shell that still remains as the appearance of a blood vessel. But it is not exactly like a living blood vessel. And that's the thing that I think people look at titles like this, and especially in the popular press, and say, oh, look, there's blood vessels there. How could blood vessels survive? Blood vessels are made of cells and all these other materials, and they have blood in them, right? So they're thinking that this is like something that's like very fresh, like. And that word is sometimes used by young Earth creationists when they describe these, like cracked open the cell and they smelled this stuff, and it was like there's fresh blood vessels there. But then you do these sophisticated analysis, and what you find are little tiny fragments of modified versions of repetitive protein pieces that form these really stable macromolecules. And you don't find all the other stuff that you would have found in cells. And that stuff would be easy to find, right? If there were actual cells there that had plasma membranes, we could extract the bilipid layer out of that, the oily layer, and characterize it and say, like, look at this. We could actually see it. I mean, to have electron microscopy pictures that would show like the individual cell walls with the organelles inside and all that stuff, right? You don't see those pictures in these papers, right? That's because all that stuff is decayed and gone long ago. It's only the really stable stuff that remains. And in this particular environment of being very protected inside a bone matrix, all right, that's what seems to have allowed for the extremely long preservation of even these fibers, which they would break down under other conditions. But once they reach a point where they're cross-linked together and pretty much have been modified to a point where they reach a point of stability, like the heme molecule reaches stability. I mean, something like heme, very similar to heme, a type of porphyrin, is found in oil. And that's been known for, many, many years, right? More than 50 years that we find these kinds of chemicals, these organic molecules in oil that we think is hundreds of millions of years old. And no one's really batted an eyelash about them surviving that long, because that cyclical aromatic compound is highly stable, right, under a lot of conditions, and can survive for hundreds of millions of years, right? So finding that particular breakdown remain of hemoglobin still present, that's not exactly shocking. And that's not the same thing as finding fresh blood in dinosaur bones. So I've used this analogy before, but I'll use it again because I think it's useful. It's like saying that there's blood vessels, like I found blood vessels in this bone, or that I found blood inside of a dinosaur bone. It's kind of like me reporting to you that I was digging in my backyard and I found $1,000 bill. I'd be like, wow, you found $1,000 bill. And this is what you envision. You're like, wow, look at that. You found this $1,000 bill. Hey, can I share in that? Let's go out and spend some of that. And you say, well, nah, actually, that's not exactly what I found. If I described it correctly to you, to give you the real impression of what I actually found, I would have to describe this. I actually found a little tiny scrap. All right. Everything else is decayed away, and there's just this little tiny bit left, but I can tell just from this little tiny bit that the molecules that are there represent something that I can identify as having been part of $1,000 bill sometime in the past. Right. And so what I actually have is just the remains of that, but I'm saying I found $1,000 bill. It's like I'm saying, I found blood, right? I found the remains of a piece of blood. It's not the same as saying we found fresh red blood cells. All right, I think I've repeated that enough times now, but I keep saying I've repeated it enough times, and yet I still see young Earth creationists constantly refer to blood being found, fresh blood being found, hemoglobin being found, red blood cells being found, intact vessels being found. All those contain a little bit of truth, but the average person hearing those words without the context will think that these are things that are not decayed, and that's not the case. All right, let's wrap it up there. Thanks for listening. We'll talk to you later. Bye-bye.